Like they'll join, ma'am. Let us start.
Oh.
Okay. Okay. So,
So yeah, Rashmi Rekha, shall I share the screen?
yes, madam. Let me introduce myself first. Then I'll introduce you. Then you share your slide.
Okay.
Okay. Good afternoon, all. I am Dr. Rasmi Rekha Sahu, Assistant Professor, Department of CSE ITER. I am the coordinator of today's webinar. I welcome you all to this weekly academic lecture 39 on behalf of Department of CSE ITR SOA deemed to be University. Today's webinar topic is protein superfamily classification using adaptive evolutionary radial basis function network. And it is my great pleasure to introduce our today's guest speaker, Dr. Swati Vipsita Madam. Dr. Swati Vipsita Madam is currently working as Assistant Professor in Department of CSE IIIT Bhubaneswar. She has more than 10 years of teaching experience. She has acquired her Master of Technology and doctoral degree from National Institute of Technology, Raulkela. Dr. Bipsita research interests are in the field of computational biology, machine learning, pattern recognition, data mining, and soft computing. She works in the area of DNA microarray, proteomics, and computational drug discovery. She has authored 36 articles, including both journals and conferences. She teaches data structure, pattern recognition, data mining, soft computing, machine learning, and software engineering to graduate and undergraduate students. Now I request Dr. Vipsita to present the webinar, and I request the participants to put their questions in the chat box or ask the question after the presentation. Swati madam, please go ahead now. Swati Vipsita madam, I think any network issue.
Madam, should we call madam? Like,
Uh, I think he's facing some network issue. Yes, Samiksha.
Shall I share the screen?
Yes, yes, madam, you share the screen.
Okay. Is the screen visible?
Yes, madam, it is visible.
Okay.
Madam, you can start now. Swati madam, am I audible to you?
For coronavirus. So it is the virus or the protein which causes this disease. So if the protein structure
Madam, your screen is not visible. And I think due to network issue, we can't listen you from the starting, from
just
the
hold on
beginning.
then. Yeah, hold on.
Yes, madam, your screen is visible now.
Okay. So I have progressed with the outline, what
Yes,
the points will be covered.
yes.
So the problem statement and its importance, objective, and the model which has been implemented to solve this problem, and the publication details. So as I've said you that what is the importance of the problem and where is the problem exactly fit in the real societal life. So the problem is proteins are the cause of all diseases. If the structure of the protein is correctly predicted, then the work of the drug analyst becomes much easier to discover new drugs. So that is the main reason why the drug for coronavirus is not yet discovered till date, because they are unable to predict the structure of the protein. The gene is undergoing a continuous mutation, and because of this mutation, the protein structure is continuously changing. So, if for one structure they are discovering a drug, it is not going to fit for the next day. So, that is the main problem with the coronavirus. So, here in the general real life problem, let it be a brain migraine disease, let it be a leukemia disease, or any gastric cancer, if the protein which is causing the disease, if the structure is correctly predicted, then the biologist try to classify this protein to a existing family. Once it tries to classify the protein to its existing family where it bears maximum similarity, then considering the features or the functionalities or the properties of that family, the drug will be discovered using the lock and key or the molecular docking approach. So this helps in predicting the protein function or structure of the unknown sequence, thus avoiding the expensive biological wet experiments in the laboratory. If we try to formalize the definition, once a particular sequence S causing disease D is classified to a superfamily Fi, the researchers can design some new drugs by trying some combination of existing drugs for family Fi. Thus, this classification problem helps the researchers for treatment of diseases by discovering new drugs. Now, what is molecular docking? This is a major foundation in the drug discovery phase. The analyst can perform molecular docking, which can be thought of as a lock and key problem. So, suppose it is if we consider as a lock and key, if we have a lock, then the key will be designed in the real complement orientation so that the lock opens and the lock closes. Similarly, assume that if the lock is the protein which is causing the disease, its complementary orientation structure is the ligand or it is the drug. So if the protein structure is correct, the complementary structure ligand is developed correctly, which is going to kill the protein, it means the drug analysts are successful in discovering the drug. So the name is the lock and key approach, or we can also call it as the molecular docking. So these days, computational approaches are used to derive the structure of the ligand or the drug. Also, they are using many softwares to design this drug where you use certain animations or use certain hit and trial approach to discover the drug. So now moving on, just a minute. Here the protein can be thought of as the lock and the ligand as a key. The ligand database can be searched for all possible orientations and conformations of the protein belonging to that superfamily paired with the ligand. Thus, the search space is reduced enormously as the protein in the given protein ligand pair is searched under a particular protein superfamily. The main objective of this work is to develop an efficient classifier to show high level of performance accuracy, but as the selection of significant features. So if you are having certain idea in the area of pattern classification or machine learning, so definitely you may be knowing that the selection of optimal number of significant features affects the performance of the classifier. So if too many features are taken, then it degrades the performance of the model. If too less features are taken, it also degrades the performance of the model. So we need to select significant features which are optimal in number so that the performance of the model is maximized. So the main focus in this work are on two main areas: feature extraction and selection and design of the classifier, which are implemented using the neurogenetic hybrid approach. The soft computing techniques are robust and possess the ability of tolerance for noise, imprecision, uncertainty, and approximate reasoning. So before I started my work, I have started using some trivial models, which are fundamental models, and then I took a decision in which model further to proceed. So the first work was on using feed-forward backpropagation. The second work was on probabilistic neural network, and the third one was on RBFN. So these three models you can see here, the graph showing the performance of feed-forward, RBFN, and PNN, and it is observed from the graph that PNN is outperforming the existing models. But still, why the focus is on RBFN? The two major drawbacks of a feed-forward neural network are very slow computing speed and the possibility of getting trapped in local minima. So it is a very fundamental model. You may be aware of it. But probabilistic neural network, although performed well, but the major drawback of PNN is it performs well when the training data set is small in size. So if I have hardly 20 tuples or hardly 50 tuples, at max 100 tuples, then this model will perform well if the data set is small in size. But as the size of the training data set increases, the architecture of PNN becomes too complex, as every node in the pattern layer represents a training sample. So if you want to can go for the detail in PNN, and the founder is Donald Specht, you can refer some tutorials on PNN, how the working is exactly done. And Donald Specht is the founder. So we prefer to, because you can see here, feed forward has its own disadvantages, PNN has its own disadvantages. So we prefer to select RBFN to classify the amino acid sequences to their superfamily. Now, the basic concept and architecture of RBFN. If you look into this model, you can see here, there is an input layer, there is a hidden layer, and there is an output layer. So it is a three-layered network. The number of nodes in the input layer is always depending upon the number of features on which you work. And the number of nodes in the output layer depends upon the number of classes you are working on. So if it is a 4-class problem, there will be 4 nodes in the output layer. If it is a 3-class problem, there will be 3 nodes in the output layer, and so on. And the number of nodes in the input layer depends on features. So the real challenge lies on how to select the number of nodes in the hidden layer, because it controls the computational complexity and the topology of the model. So it is a very big trade-off problem. If you take too many number of hidden layers, the computational complexity becomes too high. You may not work on your single machine. And if there are too less number of hidden layers, then the performance of the model may degrade. So you need to find a balance between the number of nodes, which should be optimal number of nodes in the hidden layer. So the input layer, if you look at the model, the input layer and the weight connecting the input and the hidden layer are all set to unity. The weight matrix does not undergo any updation, but the weight connecting the hidden and the output layer, that is WKJ, it undergoes a learning process. So we have 3 approaches for RBFN learning. So here I have followed the steepest gradient descent learning. And the number of nodes in the hidden layer will be taken from the training samples. The activation function in the hidden layer is the Gaussian activation function. The output of the Gaussian activation function into the weight matrix connecting hidden and output is the signal which is transmitted to the output layer. And then we have the model output. So the final output is produced by y is equal to fx, which is wi into fix, that is the Gaussian activation function into weight value. A normalized Gaussian function is usually used as the radial basis function. So the signal function which is used here is e to the power minus x minus ci. Ci are the central nodes which are fetched from the training samples, divided by 2 to the power sigma I square. x1, x2, xn denotes the input vector. C1i, C2i denotes the ith center vector, and sigma I square represents the width parameter or the smoothing parameter. So sigma is one of the controlling parameter of RBFN. If you start varying sigma, the performance of the model will vary accordingly. So structure optimization of RBFN using AMOGA. The problem of finding the number of hidden centers remains a critical issue in the design of RBFN. So as already stated, that if you are taking too many The topology of the model will be very complex. You may need GPU machines to solve the problem. And if you are taking too less number of hidden centers, then the performance of the model will degrade. So there need to be a balance between the two conflicting objectives. The number of basis function controls the complexity and generalization ability of the network. Thus, a trade-off between the accuracy and the computational complexity arises, which can be solved by selecting optimal number of hidden centers from the Pareto optimal set using adaptive MOGA. Besides the number of hidden centers, the weight matrix connecting the hidden and the output layer also affects the accuracy of the model. So the weight matrix needs to be optimal, the number of hidden centers need to be optimal so that we can derive a model which really performs better for classifying the protein sequences. So if you look at the brief overview of the entire process, and if people want to work on this domain, so please have a look at how do you start your problem. The problem is you can refer UniProt database, protein sequence database, universal protein repository, which has amino acid sequences extracted in FASTA format. So you will be having, you can extract it, you can store it in a notepad, and there are other database also, Scope, CAT. If you refer this database, you can also get the protein amino acid sequences. But every machine learning model cannot work with the categorical data. If I have these amino acid sequence, I cannot pass it directly to the model of RBFN or feed forward or PNN. So data pre-processing is a major step for any pattern classification problem. So the features which are selected from the amino acid sequences are atomic composition, molecular weight, isoelectric point, average mass of protein sequence, amino acid distribution, 2-gram distribution, exchange group distribution, and 2-gram distribution, 2-gram exchange group distribution. So these features, when I have worked, there are certain features which you require manually to code it, and certain features there are already built-in functions. Suppose molecular weight, there is already a function in the bioinformatics toolbox in MATLAB. If you pass the amino acid sequence as the parameter and molecular weight as the function, directly the function will give you a value 4.2 or 4.8 and so on. Atomic composition also there is a formula, but for 2-gram distribution, sometimes you have to write the code and fetch the features. But still, these days you have BioPython library, and if you refer to this BioPython library, there are many functions which you can directly pass the amino acid sequence as the parameter, and you can directly get the numeric value. So ultimately, the categorical data is converted into numeric data. Now, after I have derived all these features, it was observed that the matrix which was derived is a sparse matrix, which has too many number of zero entries compared to non-zero entries. So the next phase is I went for dimension reduction using principal component analysis so that I can retrieve the important features from the data is mapped to another dimension, and there the necessary principal components will help me to extract the distinguishing features and discard the non-significant one. After the PCA, after a PCA is implemented, the principal components were actually then was actually contributed in the AMOGA part. So which principal, for example, I have derived 57 or 60 principal components after using PCA, but again I can search the eigenspace using genetic algorithm. 60 has given me the value which was distinguishing, but still further to find more significant features, I have used AMOGA so that the eigenspace is explored in an efficient way, and genetic algorithm has contributed to explore the eigenspace and still give me the optimal number of features. So classifier was RBFN plus AMOGA to predict the protein superfamily, and there were 4 superfamilies in which I have validated the approach, and then the performance evaluation of the classifier was done. So this is the overall idea of the entire process. So now, if you look at this UniProt data acquisition from the database, UniProt or PubMed database, you can see the FASTA format. There will be a prompt appearing at the starting, which shows that this is a new amino acid sequence. And when the next prompt arises, it will show that it is the second amino acid sequences belonging to the superfamily. So amino acid sequences is downloaded and saved in FASTA format. Therefore, for every amino acid sequence, 470 features were processed to build the fixed dimension feature vector as follows. So 1, 2, 3, 4, 5, the column indices. These are all the information regarding the column indices. So 1, 2, 3, 4, 5 was atomic composition. X6 was molecular weight. 7 isoelectric point. 8 average mass of protein sequences. 9 to 28 amino acid distribution. 29 to 428 2-gram distribution. Suppose if it is ABCD, so it is, if it is 2 gram, just like you have your gates, logic gates, ABC. So if it is a binary combination, the base is 2. So if it is 3 gate, so you used to get 2 to the power 3 number of combinations, 8 combinations. So considering the same idea, if there are 20 amino acids, so 2 to the power 20, that is of 400 combinations of 2-gram distribution. So you can see the indices is lying within a range of X29 to X428. Similarly, exchange group distribution and 435 to 470 2-gram exchange group distribution. This set of 470 features and the training and test samples derived from 4 protein superfamilies were applied for training and testing of the model. So this is the sample input feature matrix of the protein data. So the number of rows will depend upon the number of tuples you are dealing. Suppose it is a globin database, you may have 400 tuples. If it is ribitodihydrogenase, another superfamily, you may have 1000 tuples. So the number of tuples or the sequences under study for a particular superfamily will vary from one class to the other. But the number of features, that is the column indices, was fixed to be 470. So you can see here there are a number of 0 entries. The overall matrix was, and I could not show, it is just a screenshot of the input feature matrix. So multi-objective optimization. So more than one objective is a multi-objective. So in multi-objective optimization, problems may exist where the objectives are quite conflicting to each other. Generally, these problems can be solved by making a pairwise comparison and arranging them in several non-domination fronts or Pareto fronts based on the rank. So the 2 main goals for any Pareto optimal solution, so those who are working in the domain of multi-objective GA, there are a number of strategies. Pareto optimal is one such strategy. You have weighted sum approach, you have niched Pareto approach, elitism approach. So there are a number of strategy. So the strategy which I have followed here is the Pareto optimal strategy. So the 2 main goals for any Pareto optimal solution are: the solution should converge as close as possible to the true Pareto optimal front. So this is the normal property. If you are working on a neural network, if you want your error to be 0, So you should at least approach 0.001. So you should try, or you should approach to the global solution. So if you are at least tending towards moving towards the global solution, still your approach is preferable. And the second main goal of Pareto optimal solution are the solutions should be as widely spread as possible on the best Pareto front. So when you are deriving or moving towards deriving the true global Pareto front, the solution should be uniformly scattered across the Pareto front. So if they are clustered, we need to little bit recode it so that the points need to be scattered. You have points in the extrema, and also you have certain points in the mid region. Then only the solutions are uniformly spread. So this is one of the major property of a Pareto optimal approach. So you can see here A, B, C, D, E. These I have F1, one of the fitness function in the x-axis, F2, one of the fitness function in the y-axis. When I'm deriving this Pareto optimal front set, you can see some will lie scattered like G, F, H. These are the solutions, but they're not in a Pareto front. But the global Pareto front has A, B, C, D, E. And here you can see the solutions are uniformly scattered in the global Pareto fronts. So A, B, C, D, E are actually called as the non-dominated points. So this is a schematic representation of Pareto optimality and dominated points. Basic concept of adaptive MOGA. So what is the main feature of adaptiveness and how it varies from normal MOGA? Adaptive MOGA strictly follows the concept of survival of fittest, where very good solutions having high fitness value are well protected and the solutions with poor fitness value are eliminated. So this is the idea, Darwin's theory of survival of fittest. So we try to retain the good solutions because we are in a hope that they are going to produce better offsprings. And the solutions which have poor fitness are generally eliminated from the search space. The number of solutions in the best non-domination level is the yardstick to adaptively control and manipulate the values of PC and PM. So here the idea is a simple idea that if you are close to your home, you try to slowly reduce the speed of your car. It is not that the same speed which you were driving at the NH, you move on the same speed so that you will bypass your home and you move ahead. That strategy is not followed. Rather, when you are near to your home, you try to slowly control the speed or the acceleration of the car so that you pause or you stop at your home. So similarly, when the solutions are moving towards the best non-domination level, we try to regulate the probability of crossover and probability of mutation. Had it been constant, then you are again creating a disruption in the search space. So things are moving in a right direction, and suddenly if you keep the same probability of crossover, probability of mutation constant, the good solutions again, instead of getting preserved, they get disrupted. So nothing is going to come out of it if the same strategy is followed. So what exactly adaptive MOGA does, when you are moving towards the best non-domination level, we try to manipulate, we try to control the values of PC and PM. In other words, when the opt-in Pareto front merges towards the true global Pareto front, PC and PM probability of crossover and probability of mutation values are adaptively decreased to prevent disruption of very good solutions. The idea was actually derived from a good IEEE transaction paper, that is adaptive genetic algorithm. If you want, you can also refer that paper by Ellen Patnaik at IIC Bangalore. It is adaptive GA. So I have basically referred that as one of the base paper, and I have tried to incorporate that particular feature in adaptive MOGA, and the results were really promising, which you'll be seeing in the next slides. So this is the approach. I think so the algorithm the font size is less. Anyway, if you look at step number 10, others steps are as similar as multi-objective GA, but you focus on step number 10. If L, that is number of solutions, is greater than a certain percentage, n by n is certain percentage. Suppose I have taken 100 chromosomes in my search space, and suppose it is 20 by 100. 20% of the chromosomes are at the best global Pareto front. Then I try to decrease probability of crossover PC is equal to PC minus constant step factor. PM, probability of mutation, is equal to PM minus constant step factor. So slowly, when I am checking this value, n by n percentage, how many percentage of solution are at the best non-domination level, and then I'm manipulating PC and PM. So if L is greater than or equal to a certain predefined percentage, I have taken 100 chromosomes and it is suppose 80 chromosome. I fix a threshold that 80 chromosomes out of 100 are in the best non-domination level, then termination criteria is met. I exit multi-objective GA. So coming to the experimental details and simulation results. So I have worked on 4 protein superfamily. Globin had 840 tuples. 588, I have taken them. I have maintained a proportion 70 and 30%. Training 588, test sequence 252, because you won't get uniform sequences for every superfamily. So amino acid sequences under globin, under kinase, they will be varying. So 542 under kinase, 380 was taken as the training sequence, 162 as the test sequence. Ribito dehydrogenase 828, 580 training and 248 testing. And ligase 678 total, 475 was training, 203 was the test. So details of using AMOGA, the initialization of chromosome, the population size was fixed at n is equal to 40. That is, the chromosomes are my search agent, and this search agent will try to efficiently explore the search space. Probability of crossover was initialized as 0.8 and probability of mutation was initialized as 0.008. Now, if you look at the chromosome has 2 parts. The first part of the chromosome is the number of, how will you choose the Gaussian functions? Because the Gaussian functions are actually the training tuples. And the second part of the chromosome is showing me the synaptic weight. So the genotype of the chromosome consists of 2 parts. The first part is binary encoded, which controls the topology of the network. The first part is binary encoded, so which controls, that is, the number of nodes in the hidden layer. You can see here the arrow showing G1 to G50. So here 0 indicates the training sample is not selected. There is a small typo. 0 indicates training sample not to be taken in the hidden layer, and 1 indicates training sample to be taken in the hidden layer. So these 150 chromosomes, or this 150 is actually, if I have worked on 4 superfamilies, uniformly the tuples are selected. Maybe 30, 30, 30, 30, or 35, 35, 35, 35 from every superfamily. And if there is a 1, it means that particular tuple will form a node in the hidden layer. And if it is 0, we discard that tuple to contribute in the hidden layer. And based on ones, there will be the connecting weights. Suppose I have 20 ones in the first part of the chromosome. The second part will have 20 genes. You can see here, you can see in the model, if there are 20 Gaussian nodes, those 20 are actually connected to the summation value. So if there are 30 ones in the first part of the chromosome, which is always randomly initialized, there will be 30 genes in the second part of the chromosome because they are directly one-to-one connected to the summation layer. Besides that, there is a bias neuron which is connected to the output layer, and the bias weight is generally set to unity. So we have a bias node or a bias weight to control or manipulate the model either when a network paralysis occurs or if the network toggles very fast within a specified range, then the bias neuron is manipulated to control the working of the model. So this is the distribution of the gene of the MOGA. Now in the weight extraction formula, which is the second part, G1, G2 up to GL chromosomes, which represents the gene of the chromosome. And how do we manipulate it? Suppose we take 5 digit, you can see here, if it is a 5 digit, just a minute. Hold on, just. Yeah, if we have 5 digit, we first check the first digit. If it is greater than 5, the gene weight will hold a positive value. And if it is less than 5, the gene weight will hold a negative value, and rest all is converted into decimal. So gene 1 is 7. So it is positive 0.3214. Gene 2, it is a negative value because the first digit is less than 5. So minus 0.7832. So this is the weight extraction formula, which is just written using some mathematical equation. The second part of the chromosome has variable number of genes as it depends upon the number of ones in the first part of the chromosome. Evaluation of fitness function. So the first objective function F1 is to minimize the number of hidden centers. As I have already said you, we need to find the optimal topology. And the second objective function is to minimize the mean square error of the model, which is the difference between the neural network output and the target output. So there will be a target output because it is a supervised learning. So when we are setting the target, we follow the approach of one-hot encoding. And the model is going to give the output. The difference between both is the error. The fitness function may be defined as F2 is equal to minimize mean square error, and the mean square error is Ej square n. So J is equal to 1. J is the total number of neurons in the output layer, and L is the number of training samples. So we are finding the cumulative sum to find the overall mean square error, that is the error obtained in each iteration over the total number of output node, and we cumulatively collect it over the total number of training samples. So J is equal to 1 to capital J. It is actually denoting the number of neurons in the output layer. And when it is cumulative over L is equal to 1 to L, it is the cumulative training error over the training tuples. So this is the difference between the neural network output and the target. So this is crowding distance assignment. So this has complete concept with relation to multi-objective GA. So if you want to explore it, you have to study MOGA in detail so that you can come across what exactly is crowding distance or binary tournament selection. And then you can get the idea regarding this approach. So this is adaptive crossover, which is the practical implementation details. So when the number of non-dominated solutions in the first Pareto that is absolute value or the cardinal of F1 is greater than or equal to 25%, slowly I have tried to reduce PC. PC is equal to PC minus 0.2, PM is equal to PM minus 0.002. So the probability values were updated when the number of non-dominated points in front one was more than 50%, 70%, 90% respectively. So there were 3 to 4 phases. So when it is 25%, I have reduced. When it was 50%, I have decremented the value of PC and PM. 75% I have decremented, and when it was 90%, I have decremented. So the stopping criteria, AMOGA process terminates when 90% or more number of solutions are in the best non-domination level. So the number of nodes in the input layer was decided on the basis of reduced feature vector dimension obtained after the implementation of PCA. So as I have said you that I had got an input matrix, but it was a sparse matrix. So for that reason, PCA was implemented so as to get the significant reduced dimension. So that particular PCA matrix was fed as the input layer to the model. The number of hidden nodes were selected from the mid region of Pareto front. So the solutions were extracted from the mid region of Pareto front. And every solution is a chromosome. And that chromosome was giving me two values. The first part was giving me which are the centers to form the hidden nodes as well as the connecting weight. So this neurogenetic approach is a powerful approach because it directly gives a value of the weight matrix. And the target vector for the 4 protein superfamilies are one-hot encoding. All of you may be following this approach, one-hot encoding. For the first protein family, the target vector was 1,000. For the second, it is 0100. Third, it is 0010. And for the fourth superfamily, the target was 0001. So this is the way the input, the hidden, and the output layer were fixed before the coding or the implementation began. Parameters. The parameters used for measuring the efficiency of two approaches are convergence rate and predictive accuracy. So the confusion matrix, we have the idea of true positive, true negative, false positive, false negative, true positive, positive getting correctly classified as positive, true negative, negative getting correctly classified as negative, and false positive, the output is positive, but practically it is a negative data, and false negative, the output has come negative, but practically it is a positive data. So this is the parameter to judge the efficiency of the model. Now, if you look at the results from the numerical simulation, after PCA was applied for significant feature extraction, 99% of the variance was retrieved by the first 57 principal components. So, what is the importance? If we extract 57, 99% variance from 57, so working on this 57 is as like as working on the remaining 100 or 200 or 470 components. So if it is a 470 feature matrix, I will be having 470 principal components. But 99% of the variance is already retained by first 57 principal components. So working on this 57 is as like as working on 470 PCs. So why not to work on 57 principal components, which has really given me a reduced dimension in a different domain, in a different dimension. So now, if you look here, you can see that the performance of AMOGA and MOGA when sigma is equal to 0.3. So I have varied this smoothing parameter. As I have said you that the smoothing parameter are a controlling parameter in RBFN. So 0.3, 0.5 and 0.7. So the Pareto fronts are derived based on varying. So you can see here the first one, Pareto front obtained after 355 generations of AMOGA when sigma is equal to 0.3. And here, to reach the same, only MOGA took 787. So here you can judge that AMOGA is more efficient with respect to convergence speed. Similarly, when sigma was varied, AMOGA took 1019 generations, whereas AMOGA took only 338 generation to converge. Similarly, when sigma is equal to 0.7, MOGA took 967 generations, whereas AMOGA took 460 generations. So Pareto optimal set of a first Pareto front of AMOGA after 338 generations. So you can see here that these are my non-dominated solution. 7 non-dominated solution which was fetched from the first Pareto. And let me take that, instead of taking the solutions from the either extrema, it is always wise to take from the mid region. So mid region has given me 61 number of centers, and the misclassification error, the second fitness function, is 0.0062. And here if I take from the mid region, 62 are the number of centers, but the accuracy, there is a little bit high error, that is 0.0079. So you can see here, if you take solutions from the mid region, the F1 and F2 are more efficient in AMOGA compared to MOGA, and the generations are quite less in AMOGA compared to MOGA. So you can see here performance of MOGA and AMOGA. So when sigma is equal to 0.3, number of generations was 787. From globin, 244 sequences were correctly classified. From kinase, 156. Ribitodehydrogenase, 238 tuples were correctly classified. And from ligase, it is 182. So the accuracy is 94.79. So sigma for 0.5, you can check the result. It is 96.18. And sigma is equal to 0.6. You can check the result. It is 93.87. If you look at adaptive MOGA, for sigma is equal to 0.3, the accuracy was more higher, 97.57, and convergence was within 355 generations. Sigma is equal to 0.5, the convergence was 338 generation, and accuracy was 97.91, and sigma is equal to 0.6, the number of generation is 460, and the predictive accuracy is 95.6. So if we find out a cumulative figure of MOGA and AMOGA, you can see here, feed forward BP has given 85.33, which is very low. Probabilistic neural network, 92.67. Standard RBFN, which was not hybridized with genetic algorithm, gave 84.13. The accuracy was not that good. But when RBFN MOGA was used, there was an improve to 96.18, and RBFN and MOGA still gave better accuracy, along with, you have already checked, the number of generations which it took time to converge was still better, and the accuracy was 97.91%. So from the above numerical simulations, it can be concluded that AMOGA outperformed MOGA in terms of speed and accuracy by protecting the high fitness solution from getting disrupted in the search space. The RBFN obtained from the ensemble of networks has shown good classification accuracy in comparison to standard neural networks, which is validated by performing classification considering 4 protein superfamilies. The results are quite encouraging, and some more real-life problems can be solved using this approach. And work can be further extended by developing some more adaptive concepts to improve the performance of MOGA. So this particular work was published in 2012. It was one of my contributory research work in International Journal of Computational Intelligence and Applications, 11th volume, number 4 of World Scientific Press. So this was all about the presentation. These are some standard references which I have referred to implement this work. And if you have any queries, you are always free to ask me so that I can answer and clear some of your doubts. So please move ahead.
Thank you, madam, for your nice presentation. Anybody is having any question, please ask to madam. Okay, so no questions. Actually, madam is not well since few days, but she kept our request and delivered such a nice presentation. Now I request Dr. Bharat Jyotiranjan sir to give vote of thanks. Bharat sir.
Yeah, thank you, ma'am. Good afternoon, everyone. And on behalf of the Department of Computer Science and Engineering, I would like to express our deepest gratitude to Dr. Swati Vipsita. So today's topic was very smooth, and it really inspired me to explore the interdisciplinary research. So after this COVID situation, we were all wondering how the solution is going to come out. But I am happy to know that also our the computing science field has some contribution which can help to other fields like this protein classification. So this inspire us to look into more interdisciplinary research. Thank you, ma'am, for introducing such a nice topic on the weekend evening. Once again, thank you everyone for joining. With this, I would
want
also
to have some collaboration because
yes,
we have a
yeah.
small team working in computational drug discovery. So if you want
Yes,
to collaborate with the IIIT, you are always welcome to talk to me or Anjali ma'am or
yes,
Sabir
sure, ma'am.
Sachipadhyay,
Some of
to
our
proceed with the
students are
research
working on
further.
data science. Yes,
Yeah.
some of our students are working on data science and machine learning. So
Yeah.
we'll find interested student and would like to collaborate with you, ma'am.
Definitely, we are always there.
Okay, thank you, ma'am.
Thank you, all of you.
So with this, I also extend my deepest thanks to the organizing team members. Thank you, everyone.
Rashmi